Hello, in this video we're going to deal with the Nobel Prize in Physiology or Medicine for the year 1910, awarded to Albrecht Kossel in recognition of the contributions to our knowledge of cell chemistry made through his work on proteins, including the nucleic substances. And this video will deal with his lecture. A previous video dealt with the general information, biography, work, and the award ceremony speech given to him, but this is his own lecture on the chemical composition of the cell nucleus, and it is summarized by me. And in the end, you will find the link to the complete lecture if you want to read that. The development of organic chemistry builds on concepts as the arrangements of the atoms in space. The chemical structure of organic formulation in animals or vegetable tissue is investigated just as anatomy does with the structure of living beings. All hope to gain knowledge on the function of animals and plants. Experience on the composition of the cell and the protoplasm are a step towards this. There are certain vital processes common to all plants and animals, a chemical mechanism. The main focus is today on the physiological combustion process, from which at the same time the remaining parts of the body proceeds, and especially the protoplasm. Living cells always contain products of its physiological activity. Distinction between components and, in conclusion, between organized body substances and chemical metabolism is hard to define. Results can only be expected from histologic appraisal and comparative study. New prospects were opened when the cell nucleus could be investigated. Its structure and function must be associated with the general processes of life, and this is clear looking at cell division processes which occur in animals and plants alike. Chemical characteristics on the nucleus are added to the morphological ones. Hoppenheimer and Mischer were able to isolate nuclei. They contained a phosphorus substance, a nuclein. This material proved to be peculiar to the cell nucleus. The nucleic material found belonged to a well-defined part of the nuclear substance called chromatin. A problem, nucleic substances were also found in animal products which were contained in no cell nuclei, in yolk sacs of eggs and in casein of milk. Only chemical investigation could explain this. The chemical structure of these nuclear substances so peculiarities which are bound to the protoplasm. These components break down in building blocks. The blocks fit together in a great number and variety from the molecules of protein, starch, glycogen, fat, phosphates, etc. The building blocks are made during the digestion of food and can be fitted together again. Nucleic substances show a composite of this sort also. They split into a protein and a nucleic acid. I found 4 nitrogen-containing groups of these: cytosine, thymine, adenine, and guanine. Some of these were already known to exist in sperm and originated from protein. The idea of their origin from nucleic acid met with opposition. Their presence in blood with leukemia patients. But these objections were overcome. Also, it was known that there is no nucleic acid present in yolk and milk. The question of how the nitrogen and carbon atoms in the molecule are arranged. Adenine and guanine are alloxan derivatives of purine derivatives. Scheele, Tobin, Bergen, Wöhle, Liebig, Stecker, von Baer, Fischer all did brilliant work on these substances. Fischer's work led to the formulae. See figure A. Thymine and cytosine show the similar variation. Also see figure A. Here it is. Here you see the 4 compounds. Yeah. It is evident that in thymine and cytosine, a ring-like system of C and H atoms must be assumed. The position of the atom in cytosine could be determined. It is a simple pyrimidine ring, while adenine and guanine have double purine ring with more H atoms. Thus, C and H atoms fit into the same basic plan. The purine ring arises out of a structural addition to the pyrimidine ring, and atoms can be added to and split off the ring. The atoms in the ring are difficult to separate. The NH2 ring can be detached by introducing water, giving anthroposophy, Viennemi, and uracil. There is still remainder made up of two dissimilar products, one with 6 C atoms linked with H and O atoms, hydrogen and oxygen in common, and phosphoric acid. See the picture in the next slide. What are the relative amounts? How are they mutually arranged? Still, answer this first. For each of the 4 H-rich groups, there is one carbohydrate molecule and one phosphoric acid. The first two are often found together, even if the nucleic acid is broken down. And here you see the illustration referred to here. Nucleic acid has been shown to exist from a complex of at least 12 building blocks in the living cell, maybe and even more. There is an important diversity in the structure of this class of substances. There exists much more than 4 and contain proteins, fats, bile acid, etc. The structure of the nucleic acid is repeated in other organs, but they're more simple. Nucleic acid in yeast misses thymine and has 5C carbon ring, not 6 carbon ring. Even more simple ionizes and guanylylic acids. All these and others have less nitrogen groups, 5C instead of 6 carbons in the ring. Investigations were done by Liebig, Heiser, and Hammerstein-Bang. The simpler nucleic acids are now further investigated. They are more easy to understand, and it's not clear how important they are for the life of the cell. The nucleic acids are found in the cell nucleus in combination with proteins, a large variety of combinations, behaving all like salts. Some varieties have a strong resistance to the action of chemical agents and others have not. Kossel then gives more examples, which I skipped here for reasons of time, but you can read the original text, of course. A link is in the description. The nuclei in which there is a loose combination of nucleic acids show another remarkable phenomenon. The proteins with which they combine behave like an organic base. The nuclei in which the protein is firmly combined are much less susceptible to chemical investigations. Invertebrates are loose in this respect and vertebrates are tight. In the next part, loose varieties are further investigated. I skipped it again for reasons of time. And in this part, the author points at figures B to E. Only the conclusion on the loose-bound nuclear substances is given here. Again, you can read the original, of course. If we now summarize the results of the investigations of loosely bound nuclear substances, the results are as follows. A composition of the chromatin substance of the nucleus from 2 compounds, the one which rich in bound phosphoric acid and have the qualities of an acid, the second having a protein with the qualities of a base. In the chemical structure, both components show a notable similarity based on the remarkable combination of nitrogen atoms, and because of this chemical structure, the chromatin foundations can be sharply differentiated, difficult word, from the remaining cell components, and this quality must obviously be associated with the function of the chromatin substance. These atom groups, rich in nitrogen and containing phosphorus, are those whose deposits in the chromosomes are first set in motion during cell division and whose transmission to other cell forms an essential part of the reproductive process. At this point, we have arrived at problems, the solution of which can only be attained by various methods of research working together. The representatives of morphological science see under a microscope a structure deposited in the cell and study the dependence of its form on the conditions of the elementary organism. The biochemist tries to define the composition of its structure, its position in the chemical system, and at the same time its relation to other chemical components of the cell. But this task demands theories of structural chemistry and the aid of synthetic method. Thus the results, which I have attempted to portray, today we have originated from various research institutions, and the names of many people will have to be mentioned if the merits of all contributors were to be acknowledged. Okay, this was the summary of the 1910 Physiology/Medicine Nobel Prize lecture. If you want to read the complete text, follow these links, which will also be here in the description of the video. For now, thank you for watching and the next time we will, of course, go to the year 1911. Again, thank you for watching, and maybe until another time.